Okay, so you have this incredible idea for like a groundbreaking new drug, something that could be a total game changer for a specific disease.
Mm.
But how do you go from that initial spark to an actual safe and effective treatment, something people can actually take?
Yeah, well, that's where the whole intricate world of drug development comes in. And today, we're going deep into a crucial stage, those first human trials, phase 1 trials. That's where a new drug is tested in humans for the very first time.
It's like stepping into completely uncharted territory, right? You have this promising drug candidate, but you need to figure out a lot of things like, is it safe? How does it actually behave in the body? And does it even have a chance of working?
Exactly. And to navigate those initial trials, scientists rely on this dynamic duo PK and PD pharmacokinetics and pharmacodynamics.
Okay, I'll admit those terms always sound a bit intimidating to me,
Hmm.
but they make sense once you break them down, right?
Oh, absolutely. Pharmacokinetics PK is basically understanding what the body does to the drug. It's like tracking the drug's journey through the body, how it's absorbed, how it's broken down, where it travels, and eventually how it's eliminated.
So it's like we're following this drug molecule on a crazy obstacle course through the body, seeing how it navigates every hurdle, from absorption to excretion.
That's a great analogy. And while we're tracking that adventure, pharmacodynamics, or PD, helps us understand what the drug does to the body. That's where we look at its effects on cells, tissues, organs. Does it lower blood pressure? Does it shrink a tumor? Does it reduce inflammation?
So PK is like mapping the journey, and PD is assessing the impact at the destination. But why study them together, especially in these phase 1 trials?
Well, it's all about finding that sweet spot. We need to figure out a dose that's both safe and effective. To do that, we need to understand how the drug's journey, the PK, influences its impact on the body, the PD.
Okay, that makes sense.
Mm.
Can we maybe walk through an example to see how this all works?
Sure, let's say we're looking at a new blood pressure medication in phase 1.
Mm.
On the PK side, scientists will be looking at things like how quickly it's absorbed into the blood, how long it stays there, and how efficiently it's metabolized by the liver, basically mapping its concentration over time.
So tracking the rise and fall of the drug in the body. But how do they connect that to its actual effect on blood pressure? That's where PD comes in.
Exactly. Alongside the PK measurements, researchers are monitoring the participants' blood pressure at different time points after they receive the drug, looking for a correlation between the drug's concentration and any reduction in blood pressure.
So if the concentration is high, we should see a bigger drop in blood pressure, and if it drops, the effect would lessen. It's all about connecting the PK and PD dots.
Precisely. Analyzing both sets of data helps scientists understand the relationship between the drug exposure and the effect. This helps determine the optimal dose for later trials, a dose that actually works to lower blood pressure but also minimizes side effects.
That's like fine-tuning an instrument,
Mm.
right? You adjust the dosage just right to get the desired effect without going overboard. But how do they even measure these drug concentrations? We're talking about tiny molecules in a very complex system.
That's where the incredible world of bioanalytical measures comes in, and it all starts with something pretty simple: blood samples.
Blood samples. Seems like those play a huge role in the whole process.
They really do. They're like snapshots in time, capturing the concentration of the drug at that specific moment. Scientists use some really sophisticated techniques to analyze those samples and figure out exactly how much drug is there.
So it's not just looking at the blood itself. They're zooming in to the molecular level. But what kind of tech allows them to do that so precisely?
One of the most powerful tools is something called liquid chromatography, coupled with tandem mass spectrometry. It's a mouthful, so we just call it LCMSMS.
LCMSMS.
Yeah.
Okay, that sounds pretty impressive. I have to admit, I need a bit of help visualizing that.
Think of it like a super high-tech sorting machine separating those drug molecules from all the other stuff in the blood sample. That's liquid chromatography. It acts like a molecular sieve, isolating the drug based on its unique properties.
It's like sifting through a haystack to find the needle, but on a microscopic level. Pretty amazing.
Yeah.
But once they've isolated the molecules, how do they measure them?
That's where tandem mass spectrometry comes in. It lets scientists measure the mass-to-charge ratio of ions, which is like a molecular fingerprint for each molecule.
So they're identifying the drug based on its unique weight and electrical charge.
Exactly. By combining those two techniques, they pinpoint the amount of drug in the sample, even in tiny amounts.
Incredible. It's like a high-stakes detective story, tracking down those molecules and understanding how they affect our health. But all of this happens under some pretty strict regulations, right?
Absolutely. Organizations like the FDA and the ICH have strict guidelines for phase 1 trials. It's all about the safety of the participants and making sure the results are reliable.
So it's not just about the science. It's about making sure the science is done responsibly and ethically.
Exactly. These organizations have guidelines covering everything from how the drug is made, the qualifications of the people running the trial, to how the data is collected, analyzed, and reported.
No stone unturned. But why is data quality so crucial at this early stage?
Because the data from phase 1 trials really sets the stage for everything that comes after. If the data is bad or incomplete, it could derail the whole process.
So these regulations are there to make sure the data is accurate, transparent, and trustworthy. Makes sense.
It's also about protecting the people participating in these trials. They're often the first humans to ever receive this new drug, so their safety and well-being are the top priority. And it's not just blood pressure medications where PK and PD come in. Imagine a potential new anti-cancer drug entering phase 1.
Okay, so from a chronic condition like hypertension to something as complex as cancer, I imagine the balance between safety and efficacy is even more crucial there.
Definitely. One of the biggest challenges with cancer drugs is getting it to the tumor without damaging healthy cells. It's like navigating a minefield. You need that precise path to deliver the therapy without any unwanted explosions.
So how do PK and PD help researchers chart that course through this molecular minefield?
Well, on the PK side, we're talking about understanding how well the drug gets into the tumor, how long it stays there, and if it builds up anywhere else in the body where it could cause side effects. Basically mapping its distribution, making sure it's hitting the target, and minimizing those off-target effects.
So like making sure it takes the right exit off the highway and doesn't get lost on some back road and cause trouble.
Exactly. But just getting to the tumor isn't enough. It needs to actually work once it's there. That's where PD comes in.
Right. Just showing up isn't enough. You got to make an impact.
Precisely. PD studies for an anti-cancer drug might look at things like, is it actually shrinking the tumor, slowing its growth, or even killing cancer cells? We're looking for evidence of a real impact on the disease.
Like sending in a spy.
Yeah.
You need to confirm they've reached the target and are carrying out their mission. It's fascinating how PK and PD work together for that complete picture.
It's a really remarkable partnership, and the insights from these phase 1 trials are so valuable for guiding the drug's future.
We've talked about blood pressure meds and cancer drugs. Any other examples that highlight these PK/PD studies?
Absolutely. Let's say we're developing a new antiviral drug for a new viral infection, something like, oh, I don't know, a global pandemic maybe.
Okay, that scenario hits a little too close to home these days.
Yeah.
But I'm curious how PK and PD fit in there.
Well, with an antiviral drug, PK studies would focus on things like how it's absorbed, distributed throughout the body, if it reaches the target cells where the virus is replicating, and how long it stays active.
Similar to the cancer drug example, making sure it gets where it needs to go and stays there long enough.
Exactly. But with antivirals, you're also looking at how it interacts with the virus itself. Does it block the virus from entering cells? Does it stop it from replicating? Those are PD questions.
So not just tracking the drug, but understanding how it engages with the enemy, so to speak.
Right. The PD studies would measure things like how well it stops viral replication, reduces viral load, and ultimately helps the patient recover.
Connecting the drug's journey through the body, PK, with its impact on the virus, PD.
Exactly. And by carefully analyzing both PK and PD, data scientists can start to see how effective the drug is, optimize the dose, and identify any potential safety issues.
You know, it's easy to think of drug development as this straightforward process, but it sounds like there's a lot of back and forth, a lot of fine-tuning going on.
Absolutely. It's iterative with insights from one stage informing the next, and PK/PD studies are a big part of shaping that journey.
Okay, so we've seen some examples of PK/PD studies in action, but I'm still curious about those bioanalytical techniques you mentioned. How do they actually measure those tiny drug molecules?
That's where the science gets really exciting. Remember those blood samples we talked about?
Yes, they seem to be a key player in all of this.
Scientists use them to measure the drug concentration in the plasma. They use some pretty incredible techniques like that liquid chromatography coupled with tandem mass spectrometry, LC-MSMS.
LC-MSMS, right? That high-tech sorting machine and molecular fingerprint analyzer still sounds pretty futuristic to me.
It might sound like sci-fi, but it's a real and powerful tool. It helps us understand how drugs work in the body. To simplify, the liquid chromatography part is a separation system. Imagine a microscopic racetrack where molecules compete each with their own speed and agility.
So they're separating the drug from everything else in the blood based on how it moves through this molecular obstacle course.
Exactly, like sorting candies by size and shape. Once they've isolated the drug molecules, tandem mass spectrometry comes in. It measures the mass-to-charge ratio of ions, which is like a fingerprint for each molecule.
So each molecule has its own ID card.
You got it. Combining those two techniques lets scientists accurately identify and quantify the drug in the sample, even if there's just a tiny bit.
It's like they're doing a microscopic forensic investigation with cutting-edge technology, tracking down those molecules and understanding their every move. But this all happens under strict regulations, I assume.
Of course, organizations like the FDA and the ICH have those strict guidelines for phase 1 trials. It's all about protecting the participants and ensuring data quality and reliability. Think of it as a detailed rulebook for the whole trial.
So it's not just the science, but making sure it's done responsibly and ethically. What do these regulations cover?
A lot of things, from how the drug is made, the qualifications of the people running the trial, to how the data is collected, analyzed, and reported.
Wow, they're not messing around.
Yeah.
But why is data quality so crucial in these early trials?
Because the data from phase 1 really lays the foundation for the drug's entire future. If the data is unreliable or incomplete, it could derail the whole thing.
So these regulations are about making sure the data is accurate, transparent, and trustworthy. That makes sense.
And it's about protecting the people in the trials. They're often the first humans to receive this drug, so their safety and well-being are the absolute priority.
It's amazing to think about all the detail and precision in these early trials. It's like they're building a house brick by brick, making sure it's solid before anyone even steps inside.
That's a great way to put it. And just like building a house, there are always new ways to innovate and improve the process. We've been talking about measuring drug levels in the blood. But what if we could track a drug's journey through the body in real time without even having to draw blood?
Wait, really? Is that even possible? Sounds like something out of Star Trek.
It might sound futuristic, but it's getting closer every day. There are new technologies being developed that could let us monitor a drug's movement without being invasive. Imagine tiny little biosensors implanted in the body that could continuously measure drug concentrations in specific tissues or organs.
So instead of relying on those snapshots from blood samples, we'd have this constant stream of data showing us exactly where the drug is and how much is there, like a GPS tracker for drug molecules.
Precisely. That kind of real-time monitoring could completely change how we understand PK/PD relationships. We could see the drug interacting with its target in real time, adjust doses much more precisely, and even maybe personalize treatments based on how each person responds.
This is incredible.
Yeah.
How far off are we from actually using this kind of technology in drug development?
There are still challenges to overcome, but the progress is really exciting. Scientists are working on making these biosensors sensitive, reliable, and biocompatible, meaning they won't harm the body. And with all the advances in nanotechnology and wireless communication, the idea of real-time drug monitoring is becoming more and more realistic.
It's amazing how technology is changing how we understand drug development. It feels like we're constantly pushing the boundaries of what's possible.
It really is an exciting time to be in this field. I think these advances will eventually lead to treatments that are safer, more effective, and more personalized for patients.
Thanks to this deep dive, I have a whole new appreciation for the world of drug development. From those first steps in the lab to these crucial first human trials, it's an amazing journey.
I agree. It all boils down to this collaboration between scientists, clinicians, regulatory bodies, and of course the brave volunteers who participate in these trials. They're the unsung heroes paving the way for new treatments.
It all begins with that simple question. How do we turn a scientific idea into a medicine that can help people? And that question keeps driving innovation and discovery in this field.
Well, on that note of scientific wonder and the potential for new treatments, we'll wrap up our deep dive into the world of phase 1 trials and PK/PD studies. We hope you enjoyed the journey as much as we did.
It's been fascinating. And to our listeners, stay curious. Keep asking those questions and never stop exploring the world around you. Until next time.